You're probably here because you read the article Signs that SARS-CoV-2 is evolving to avoid the immune response. And in this video, I'll be talking about the study that supported this article and certain pieces of information within the study that I believe are really important to explain the main findings of the article that you guys read. And the main finding was that mutations are occurring in the SARS-CoV-2 virus, and it is for us to understand if the pace in which these mutations are occurring should be an alarming situation or not. Now, researchers came across an immunocompromised cancer patient, and this was basically where the research began and their findings, because after they pulled out the viral strains from the immunocompromised cancer patient, they found something really weird. One, the co-infection lasted for 2 months in the infected patients, so they studied the viral strains and found 2 different viral strains that seemed to be really different from all the viral strains that they usually came across. And what was different is that these 2 viral strains had a section of the genome that was missing. And this is called the deletion variant of the viral genome in the SARS-CoV-2 and what is basically changing. Now, mutations aren't common in SARS-CoV-2 because it has a really good mechanism of fixing itself using an enzyme called RDRP. But while fixing is one situation, it can't really notice when something is deleted. It can't really control that. So now the researchers had to ask a question as to why these deletions were being kept around. So what they did was they decided to create a copy of the deletion variants of the spike protein, and they put that aside, took a bunch of antibodies from the human body, and they had both of these entities react with each other. And what they found was there were 4 areas where deletions were really common within a section of the viral genome. And 2 of these areas, when they occurred, seemed to block the binding of the antibody, while 2 of the areas where the deletions occurred didn't block the antibody. So is this a case where we should be worried that there is a small percentage of these deletion variants protecting itself from the immune response? Yes, but it doesn't occur enough to where the immune response can't overcome with its large quantity and differentiation of antibodies. Now, the researchers of the study that supported the main finding of the article that you guys read had to go through 150,000 genome sequences to find the relevance and the repetition of these deletion variants. And all these sequences have the common or generalized formation of an NTD region, RBD, and S2. These are all parts of the spike protein. And this section is part of the longer viral genome. And in this small section, the NTD region, this is where they found 1,100 sequences, all having deletions in this area of the NTD region, this area, and this area. And that's put together as RDR1, RDR2, RDR3, and RDR4, or recurrent deletion region 1, 2, 3, and 4. The frequency of deletions were found to be mostly supported in these regions, again, because they're maintaining an open reading frame. And by that, an open reading frame basically allows a complete formation of the protein despite these deletions occurring, and wasn't found in other regions of that genome sequence, and probably why these were occurring so many times. Again, it is the non-terminal domain of the spike protein where these mutations or deletions are occurring. I said RDR1, RDR2, RDR3, and RDR4. The NTD region is specified on the spike protein as this part of that protein. The antibody will come and try to attach to either this spot as an epitope, either at this spot or at this spot. So there are other antibodies that can come and attack from this area and this area. The 4A8 monoclonal antibody is what is specified for this specific region. They have issues with the RDR2 and RDR4 deletion variants, but the RDR1 and RDR3 are successfully neutralized by our monoclonal antibodies. So, in conclusion, our immune system does have a great control over these mutations for the time being. But with time passing, as more mutations do occur, this situation where the binding does not occur anymore will cause issues with us fighting off infections, as it happened in the immunocompromised cancer patient that the researchers first saw this situation occur in.